Okay, today we're talking about the cell membrane. And the cell membrane is a structure that encloses all cells. So all cells have cell membranes. Some cells have cell walls, some cells don't have cell walls. Some cells have nucleus and some cells don't have a nucleus, but all cells have a cell membrane called the plasma membrane. So the term plasma membrane, the term cell membrane, they're pretty interchangeable. What we're focusing on when we talk about the cell membrane, the plasma membrane, is that it is made up of substances called phospholipids, and that phospholipid gives it its structure and it gives it the properties that allow it to function well. It is similar to other membranes found within the cell. So it's similar to the nuclear membrane. It's similar to the membranes surrounding the mitochondria. They're all made with phospholipids. They're bilayers. But the plasma membrane has some structures in it that other membranes don't have. The differences result from interaction with the extracellular world. So the plasma membrane is separating the cell from the extracellular fluid that bathes all of the cells in a living system. It is very thin. It is fatty in composition, which means that it's made with phospholipids. Okay, and it's stable in sight of movement of the components, the materials going in and out of the cell, and the movement and the components of the materials that actually make up the plasma membrane itself. It is made up of a phospholipid bilayer, so there are two layers, and they have cholesterol inside, and the cholesterol helps with membrane stability with changes in temperature. It has proteins inside that are essential for transportation of materials into and out of the cell, and it has a glycocalyx. That is important for recognition of cells to cells with communication and kind of stickiness, and does a whole lot of things. And what we have here is a micrograph, and a micrograph is just basically a picture taken through a microscope, and in this case we're looking at a bilayer. You can see, especially like right here, where there are two layers in this plasma membrane. All right, and so it's showing us that bilayer. And this is an artist's rendering of the same thing. And you can see all the different structures that help to make up that phospholipid bilayer. You've got two layers of phospholipids. Remember, your phospholipid has a head and two tails. And so the bilayer is set up so that the tails are on the inside. If you look right here, the tails are on the inside and the heads are on the outside. And that gives the cell membrane a lot of different properties. We have proteins that will span the cell membrane. We have carbohydrates that are attached, and they kind of act as communication and also help with stickiness to other things. So there's just a lot of components that make up that cell membrane. When inside you have cholesterols, and those cholesterols are going to help with membrane stability when it gets too hot or gets too cold. It helps to keep that membrane at the composition that it needs to be. We don't want our membrane melting if it gets too hot. We don't want our membrane to become solid if it gets too cold. So those cholesterols are kind of like the Goldilocks molecule of the membrane. It helps keep that membrane just right so that it has the flexibility that it needs in order to function correctly. All right, so let's talk about phospholipids. Phospholipids, as we talked about when we talked about lipids a couple lectures ago, are made up of two fatty acid chains bound to a phosphate group. So our diagram, like we did just a second ago, you have your phosphate group and then you have two fatty acid chains. All right, and let's actually add another page so that we can just draw our phospholipid. So we have our phosphate group, and then we have two fatty acid chains. Right there. And right here we have fatty acids. Now, the makeup of the phospholipid gives it almost a dual nature. right? Because the phosphate group is charged, it has a charge, the head of the phospholipid is polar. Now, our fatty acids are lipids. They're made of hydrogen and carbon, and they are nonpolar. Now, if you remember back to when we first talked about this, polar molecules will interact with water. So the head of the phospholipid is hydrophilic. All right, so water loving. The tail of the phospholipid, because it's nonpolar because it's a fat, is hydrophobic. So it does not want to be around water. So the structure of the phospholipid itself gives it a dual nature, where one part is okay with being around water and the other part is not okay with being around water. So when they arrange themselves so that the head is exposed to water while the tail is protected. So if I was to take this phospholipids and put them in a beaker, right, filled with water, they're going to arrange themselves so that the heads of the phospholipid are protected and the tails are all on the inside. So the water-loving heads are on the outside and the hydrophobic tails are all protected in the middle. This is actually called a micelle arrangement. If we look at our diagrams of our cell membrane, you can see where the heads are all exposed to the extracellular fluid, which has a lot of water in it, and they're also exposed to the intracellular fluid, which has a lot of water in it, but those hydrophobic tails are protected from the water. They're tucked inside. Right? So the two layers of the phospholipids are arranged with the fatty acid chains pointing towards each other. This gives, the phospholipids give the bilayer a fluid-like nature. Depending on the types of fatty acids on the phospholipid, they're going to be fluid at specific temperatures. So if you have a lot of unsaturated fatty acids on your phospholipids, so a lot of kinked fatty acids on your phospholipids, they're going to be liquid at room temperature. If you have a lot of saturated fatty acids, unkinked, they're going to be solid at room temperature. And so you can have fatty acids that are made of both. All right, so depending on the composition of the phospholipid, the cell membrane could be more fluid or more solid. And this is important because the parts of the membrane, the proteins and things like that, they need to be able to move around and migrate to where they need to be. And if the membrane was too solid, they wouldn't be able to do that. And if the membrane was too liquid, it wouldn't be able to hold everything in. So when it's made up of both saturated and unsaturated fatty acids, it kind of gives it that Goldilocks just right component. Because the tails are hydrophobic or nonpolar, only other hydrophobic and nonpolar substances can move through. So we're only going to let materials move through this cell membrane into the cell if they are also hydrophobic or nonpolar, right? So things like steroid hormones and fatty acids and things like that can cross directly through the cell membrane. But ions and amino acids, anything that's going to have a charge or is too large is not going to be able to move directly through the cell membrane. Water is one of those things that breaks the rules. Water will move through what is called an aquaporin protein. It's going to move through a channel. So remember, water is charged. So it will move through a channel to get into the cell, but water can also sneak through the cracks. Since it's such a small molecule, it kind of wiggles in through those phospholipid. It's kind of like sand in your bathing suit. Sand is not supposed to get into your bathing suit, but it can sneak through. Okay, so this is just another diagram that helps to explain what you see. So the if it is a hydrophobic molecule or a nonpolar molecule, it can move through. If it is a charged molecule, a hydrophilic or a polar molecule, it gets repelled by those phospholipids. It does not want it to go through. Something else that makes up cell membranes are proteins. So the majority of the cell membrane is that phospholipid bilayer, but proteins play a very, very important role in cell membranes. There are two types of proteins. You can have integral proteins and peripheral proteins. So if we have a cell membrane, right, and we got lots of cell membranes, heads, and we got tails. An integral protein is going to go into that cell membrane, and it might be a channel that materials can go into and out of. All right. So if the protein is actually in the cell membrane, so like it is built into those hydrophobic or to those phospholipid bilayers, it's called an integral protein. If it is to the side, on either side of the membrane, so maybe you've got a protein right here, right? Or maybe you've got a long stranded protein right here, all right? That is called a peripheral. A peripheral protein is on the side. An integral protein is built within. And they do so much things for our cells. They provide structural support, such as like this one right here is going to provide support. You can see these proteins inside the cell. You can sometimes see them outside of the cell too. Recognition, like hello, my name is. So proteins will extend from the plasma membrane so that other cells around it can recognize what type of cell it is. And that kind of leads to our communication, where receptor proteins will receive molecules like ions or compounds, and it will bind to the receptor protein, and that triggers a reaction within the cell. And then, of course, transport. And that's what you see right here. This is a transport protein that acts as channels, tunnels, or pumps that move materials into and out of the cell. So this is some diagrams that just illustrate some of this. So this is, right here, an integral protein because it is going into the cell. These are all integral proteins, right? And then this dude right here is a peripheral protein because it's to the side. This one is also peripheral because it is to the side. Some integral proteins can cross all the way through. If they can cross all the way through, like these two, they are called transmembrane proteins. Okay, and they all have very, very important jobs. Like the first one is structural support. So these peripheral proteins will bind to proteins inside the cell and help to give the cell some structure. Remember, it doesn't have that internal or external skeleton. It's just got the protein structure. Recognition, so materials are coming in and our cells are recognizing those materials. Communication, so maybe I'm sending like a neurotransmitter from neuron to neuron, and this neurotransmitter is sending information to this next neuron that, hey, maybe we need a muscle contraction. And then also transport. So maybe I need more water in the cell, or maybe I need more sodium ions in the cell, and it will go through channels. Typically, the materials that are moving through channels are either charged, and they can't go directly through the cell membrane, or too big to go through the cell membrane, like glucose. Glucose is way too big to fit through that cell membrane. All right, so we've talked about phospholipids and we've talked about protein. Now let's talk about cholesterol. Now, cholesterol, we have a tendency to think cholesterol is bad. It makes us sick for our heart, but we have to remember is that there's many different types of cholesterol out there. You've got good cholesterol, you've got bad cholesterol. Cholesterol is an important precursor to hormones, so that will develop into hormones within our body, and it's also an important part of our cell membrane. It's found in between the phospholipids in the bilayer, and what they do is that they help to maintain fluidity. It keeps that membrane from becoming solid at low temperatures, and keeps them from packing in tight, and it also keeps the membrane from becoming too liquid at high temperatures. We don't want all of our cell membranes to melt when we get hot. We want our cells to stay together so that our bodies can function. Cholesterol helps with that. We also don't want our cell membranes to become solid. We want the proteins that are important to our cells to be able to migrate to the area of the cell that they need to be in. And so that cholesterol is important because it helps to maintain fluidity. And then we have our glycocalyx. Glycocalyx will protrude from proteins and phospholipids, and they are carbohydrates. That prefix glyco is like glucose, sugar. And they're building sites for signaling molecules, so they will help to signal other cells that things are going on. They allow for lubrication of the cells so that they can move past each other, but they also allow cells to stick to each other. So they have a lot of different functions depending on what you're looking at with that glycocalyx. All right, and our glycocalyx, if you look, is right here. And in this case, it's attached to a protein. And in these two cases, let's change colors. These two cases, they're attached to phospholipids. And then we've got our cholesterol, and our cholesterol helps to maintain fluidity. And then we have our proteins, and our proteins give us structure, recognition, and movement of materials. And you can see this is another micrograph of two different cells. In fact, you have cell number 1. I don't need to cross that. And cell number 2. And in between, you've got 2 cell membranes, right? Here's a cell membrane here. You can see there's 2 layers. You have another cell membrane over here, 2 layers. And then in between you have intracellular space. And that intracellular space is filled with fluids. And that fluid is important because that's how we're getting our ions, that's how we're getting the minerals, that's how we're getting like the materials that our cells need in order to function. Amino acids, nucleic acids, sugars, proteins, things like that all get to our cells through that intercellular fluid, or that extracellular fluid rather. All right. And when we talk about cell membranes, we have to talk about what's called the fluid mosaic model. All right. Fluid as in we have movement. Mosaic meaning that we have a whole bunch of different pieces. So a membrane forming the outer boundary of most cells is composed of a phospholipid bilayer that is interspersed with proteins and cholesterol, all right, and it has carbohydrate change. So these are the pieces, the mosaic of our model. The components, all of these components, the proteins, the cholesterol, the phospholipids, the carbohydrates, have a great deal of freedom of movement. This is the fluidity of our model. Lateral movement is most common. So if you have phospholipids, sometimes they'll just kind of do a little dance and flip sides. So this one, the first one and the second one will switch, so that results in 2 different arrangements. So they'll flip sides. Proteins will move around. They'll migrate from side to side within the cell membrane. You will occasionally see vertical movement where they'll flip like this, but it's extremely rare that you see them flipping like this. Proteins, they can be fixed or they can move like icebergs. So they can move to the area of the cell that maybe if you have signaling, maybe they're going to move to that area of the cell so that you can signal more. Or maybe if a cell needs more materials, it's going to move so that it can do that. Now, some of them will do that, not all of them. Some of them will do that. But the idea of the fluid mosaic model is that the cell membrane is not a static structure. It changes according to its needs. It has a lot of different integrated parts, and those parts are all important for it to function. Now, how do we get materials into the cell? The structure of the cell membrane is such that it is selectively permeable, which means that some things can come in and some things cannot. So then the question then becomes, if it's so selectively permeable, what is allowed in and what is not allowed in? And we've already talked a little bit about this, where when we're talking about moving directly through that cell membrane, only small nonpolar materials can easily move directly through the phospholipid bilayer, and the rest of it has to move through protein channels. But we're going to specifically talk now about the different types of movement, starting with diffusion. When we talk about diffusion, we have to talk about particle motion. All particles have movement. If you are a solid particle, like a solid material, your molecules, let me make this a little bit bigger. Your molecules all have movement, right? The difference is that movement is not very big. They're just kind of sitting there vibrating, shaking in place, all right? They're the people at the dance that just kind of shift from one foot to the left foot to the right foot. They don't really get any movement. They just kind of rock back and forth to the music, right? That's kind of what a solid does. Those particles are there. They're tightly packed together. They just kind of vibrate in place. They never really move away from each other. Now, if you are a liquid, we have particles in liquids the particles in the liquids, not only can they move around, they can move around each other. So they'll shift past each other, right? They occasionally break through, but mainly they're just moving past each other, right? This would be kind of like ballroom dancing, where the couple is moving around the ballroom, in and out of other couples, but they never really break free. And then we have gases, and gases have so much energy. The molecules in gases have so much energy that they can just shoot anywhere. They move all over the place, right? They fill the container that they're in, and that's how the joy of helium balloons is that gas fills that container and it's lighter than air, so it goes up. All right. So all particles have motion. So what's the point of this? All particles have motion. Liquid particles want to move from ordered states to disordered states. And so what that means is that they're going to stay together, but they're going to interact with each other. Okay. And if we add a drop of dye, like food coloring, to water, the ordered state is that droplet is going to spread out past and move as fast as it can so that it fills the entire area to the disordered state. All right? So it's kind of like bumper cars, and that's kind of what's driving this whole process. When you're riding bumper cars, the more people riding those bumper cars, the more bumping you have, right? So if you've got 40 people out doing bumper cars, you're going to be bumping into people left and right. You're just going to be shoved all over that place. And if you are doing bumper cars and there's only 3 people out there, it's going to be really easy to avoid being bumped. You're not going to interact with those other 2 bumper cars as often as you would if there was 40 bumper cars out there. And that's kind of what's happening with diffusion. molecules in the initial drop of dye in that ordered state are going to bump against each other, and they're going to bump away from each other, and they're going to move as far as they possibly can into the container until they've reached that dispersed or disordered state. When we talk about diffusion, we're moving from regions of high concentration, like the drop of water or drop of dye, to regions of low concentration, like the water. So we want that drop of dye to spread out all the way across the water. And if we look at this diagram, it kind of explains it a little better. When we put the dye in, all of these little dye particles right here are going to be bumping up against each other. All right? Eventually, they're going to bump so far up against each other that they're going to start to spread out. And they will continue to spread out until they are equally distributed all the way through this container. Right? And once it reaches that, it's called equilibrium. Now there's several different things that can affect how fast this occurs. For instance, the more dye I put in, the faster diffusion is going to occur because you have a high concentration moving to a low concentration. The greater the difference between high concentration and low concentration, the faster diffusion goes. Another thing that can affect how fast diffusion moves is temperature. If the water and dye is at a high temperature, those molecules, remember, they're heated, they're going to have more energy, they're going to have more movement. That means they're going to bump against each other faster, and they're going to spread out faster. So increased temperature increases the rate of diffusion. Decreased temperature decreases the rate of diffusion. All right. When I talk about concentration gradients, I'm talking about the difference between high and low concentration within a given medium. All right, so if our medium is our beaker in our last slide, if this is our medium, the area where all the dye is high concentrated dye. And then low concentrated area, in this case, is going to be all the water surrounding it that doesn't have any dye. Now, natural tendency of motion is for materials to move down the concentration gradient, or rather it's going to move from high concentration to low concentration. And this is the whole concept of passive transport. Passive transport uses the energy of the material to move. It does not need added energy. It's passive. No added energy is needed. We're just using the energy that the molecule has because all molecules have energy and are moving. It is possible to move up the concentration gradient or to go from low to high, but it requires additional energy. Molecules do not naturally want to move that way, so we have to force them to move that way. And this is called active transport, and it uses ATP to power that. All right, now let's just talk about concentration gradient just a wee bit more. All right, let's see here. Imagine we're talking about a concentration gradient kind of like a stairway. We have red materials. Okay? Now, if we are going from high concentration to low concentration, from high to low, it's easy to go down that stairway, and that is passive transport. Now, if I want to go from high or low to high, if I want to move up the stairway, I have to expend energy, and that is active. All right, it's easy to go down a stairway. We can fall down a stairway, but we have to work to go up the stairway. So when we go from high concentration to low concentration, that's down our stairway and it's passive. And we could tell that this area has a high concentration of red. And we can tell that this area has a low concentration of red. So naturally, molecules want to move from high to low in passive transport, but sometimes we need them to move from low to high, and we use the process of active transport. Now, when we're talking about osmosis, we're talking specifically about the diffusion of water through that semipermeable membrane. So we are specifically talking about only the movement of water. So that membrane limits what can cross through and what cannot cross through. If the membrane is impermeable, absolutely nothing can go through. Right? If a membrane is semi-permeable, it means that some things can go through, but not everything can go through. And then there's also freely permeable, where anything gets to go through. So when we talk about impermeable and semi-permeable, or specifically semi-permeable, that means that water can go through the membrane, but maybe not all materials can go through the membrane. So maybe in our diagram's case, salt cannot go through our little membrane right here. Only water can move. All right, so if we add salt to this solution, that means there is a higher concentration of water over here because there's nothing in there. There's 100% water over there. And we added salt over here, so maybe there's only 80% water over here. So this is our high concentration, and this is our low concentration. And water will want to move over in order to try to get to equilibrium. So it's going to try to move so that it's the equal concentration on both sides. So it's trying to attempt equilibrium. And the process of water moving in order to get to equilibrium is called osmosis. Now, what this does is it can change the amount of water in a particular area. So for instance, our level of water here and our level of water here are two different levels. We've increased our water level in an attempt to reach equilibrium. That increase in water level can cause pressure, especially if it's like a closed system, if it's a system like a cell. Think of it like a water balloon. If I add water to a water balloon, I'm increasing the amount of pressure on that balloon. And I can increase it to the point where the balloon will actually rupture and it will burst. And the same thing can happen to cells. So water will move into and out of the cell. And if it gets enough of it inside the cell, the water can actually rupture that cell membrane. All right. So when we talk about semi-permeable, it means that somewhat permeable to water and liquid substances, but it is not permeable to larger substances or substances that carry a charge. So osmosis is the movement of specifically water. We're not looking at the movement of solid with osmosis, we're looking at the movement of water. And this is how organisms such as plants are able to obtain water, because we're moving it into that plant and trying to reach equilibrium. It's extremely important for metabolic processes in animals, because if we want to be able to have our cells build structures, build proteins and nucleic acids and things like that inside the cell, we have to have water in order to do that. If there is an imbalance, if the pressure, the change in the cell because water is leaving the cell, or because too much water is added to the cell, it can cause problems. Plant cells are a little bit different because they have that cell wall. They can handle more fluids than animals. They will expand until the membranes push against the cell wall and create pressure. It's called turgor pressure. In fact, this is where plants want to be. This is how plants stay standing. If plants lose too much water, they will wilt. And that wilt is because there's not as much water inside the cell, causing them to stand up. Animal cells are a little bit different though. If too much water is added, they will rupture or lyse. All right, so when they're breaking, they are lysing. And different types of water concentrations will lead to these different conditions. A fluid that has a higher concentration of solutes than the concentration of solutes inside a cell is considered hypertonic. So if I place a cell in a hypertonic solution, the water inside the cell is going to want to leave, because we're trying to reach equilibrium. It's going to try to move to that high concentrated solution outside of the cell in order to reach the same concentration of water inside and out. If I have two fluids with equal concentration of solutes, then we are isotonic, which is the same. If I put a cell in an isotonic solution, there's not going to be net gain. Water will move in and out of the cell, but the overall cell size is going to be the same. All right. If I put a cell like a hypertonic, all the water is going to move out and the cell is going to shrink. If I put a cell in a fluid that is hypotonic, there's a low concentration, all the water is going to go into the cell. And this is the case where you might have a cell rupture or break. Plants like to be hypotonic. Animal cells, if they're hypotonic, they may break. So this is diagrams that help illustrate this. Okay, so if a animal cell is placed in a hypertonic solution, that means that there's less water outside than inside. Water is going to leave. All right, and you can see water is leaving that plant, and the plant is wilted. So let's just diagram hypertonic solution real quick. We got ourselves a beaker here with water, and let's say we've got ourselves a cell in there. Hypertonic is a situation where you have a high concentration of solute or a low concentration of water. All right, so let's say that this is just 60% H2O. Right, that means that it's 40% solute. And the cell has, let's say, 85% H2O. There's a higher concentration of water inside the cell, so it wants to leave, and it's going to shrivel up. If you poke a hole in that water balloon, water is going to leave, and the water balloon is going to shrivel up. That's what happens in a hypertonic situation. Next, we have isotonic. In an isotonic situation, the concentration of water outside of the cell is the same as the concentration of water inside the cell. All right? So water is going to continue to move both inside and outside of the cell, but it's going to move at the same pace. So if we illustrate isotonic, we have our beaker with water, and it has a cell, and we have a 86% concentration of H2O outside the cell, 86% concentration of H2O inside the cell. So water is going to go out and it's going to go back in, but there is no net change. And that is what is called an isotonic solution. No net change in cell. Where hypertonic is the cell loses water. That's a 2. And shrinks. All right, so hypertonic, isotonic, and what is left is hypotonic. In a hypotonic solution, there's a greater concentration of solute inside the cell, so water wants to move in. That's our balloon. If we overfill our water balloon, and we're talking about animal cells, they're going to rupture, they're going to bust. If we're talking about plant cells, this is their happy place. Plants want to be hypotonic because that means they have all the water they need in order to function correctly. All right, so hypotonic. One of the ways I tell my students in my classroom is to remember that. This is that cells in a hypotonic solution get fat like hippos. Hypo, hippo. Cells in a hypotonic solution get fat like hippos because there is a greater concentration of water outside the cell than that's inside the cell. So water moves in order to try to equal that out. So the cell increases in size and can burst. All right, so what is the main difference between passive and active transport? Passive transport is any movement of molecules or ions across the cell membrane that doesn't require expenditure of energy, or ATP. So we're moving from high to low, and examples are going to be simple diffusion and facilitated diffusion. A facilitated diffusion is diffusion through a protein channel. So it's still diffusion, it's still moving from high concentrations to low concentrations, it's just moving through a protein channel, such as water through an aquaporin or glucose through a transport protein. Transport proteins are very specific. They will transport a specific class or a specific type of molecule, and only that particular class or type of molecule. That they will actually bind to the protein, and that binding can change the shape of the protein, allowing it to push that material in. If it's just a channel, they're not as specific. Basically, it's kind of, if it fits, it ships sort of situation. If the molecule can fit through the channel, it's going to be able to move. And so facilitated diffusion is still diffusion from high to low concentration, but we're just going through that protein channel. Active transport, however, requires the expenditure of energy. ATP is used, and the example of an active transport is the sodium-potassium pump. And what we're doing with active transport is we're moving against the concentration gradient. So we're moving materials from low concentration to high concentration, and this is not where they naturally want to move. They want to naturally move the other way. So simple diffusion is movement of materials through that bilayer directly. Facilitated diffusion is the movement of materials through a protein channel, and then active transport is the movement of materials through a protein, but it's moving from low concentration to high concentration. All right, and it's using ATP to power that movement. So specifically talking about the sodium-potassium pump, we're moving sodium and potassium ions, Na plus and potassium plus ions, through active transport. And it allows us to maintain high concentration of potassium ions inside the cell and high concentration of sodium ions outside the cell. So we're wanting to make sure that there are more potassium ions in the cell, and they're wanting to make sure there's more sodium ions outside the cell. And you might say, why are there situations where you don't want it to be equal? Well, when we maintain that concentration gradient, especially when you're talking about an ion, it helps to maintain an electrical gradient. So we're looking at the charges, and it helps to maintain charges. And if you're talking about something like the nervous system, it helps to send signals. The movement of those ions, that electrochemical gradient, allows for signals to occur within the nervous system. So ions, in this case, ions want to move with the respective concentration gradient. Potassium wants to move out, sodium wants to move in, but the sodium-potassium pump maintains the concentration gradient. So you see these a lot in nerve cells, and it allows for the conduction of nerve stimulus. So here's how this works. Naturally, sodium wants to move. Hold on, I just forgot it. Sodium wants to move in, but we're constantly pumping sodium out. And as we pump sodium out, that allows us to move potassium in. All right. So the binding of the sodium atoms to the potassium pump causes it to change shape, and that allows that sodium to be pumped out. And it also allows the potassium to bind in. This is the same pump. It's just when in one configuration, sodium binds, and in another configuration, potassium binds. All right, so we've talked about how do we move ions, how do we move materials, but some materials can't even go through a protein channel. Some materials have to be brought into the cell in other ways. Exocytosis is the movement of materials out of the cell through a vesicle. All right. So we might have inside the cell old cell parts, old cell waste that we want to get rid of. Maybe we're wanting to release a bunch of proteins so that they can be moved to a different portion of a system. So what happens is these molecules are too big to directly cross. All right, they may even be too big to move through a protein channel, right? So we have to find a way that we can get them outside of the cell. This is called exocytosis. You just remember exo equals exit. So this is we're moving materials out of the cell. This vesicle right here is actually going to fuse to the cell membrane. And when it fuses to the cell membrane, that allows us to open the vesicle so that the materials can leave. And so if you notice, the cell membrane never exposes the inside of the cell, right here, to the outside of the cell, right here. We bind this vesicle so that we can then pinch it open and allow the materials to move out. Exocytosis. All right, and this isn't a micrograph. So this right here is our cell membrane, right? This is our vesicle. You can see where it's bound directly to the cell membrane, and then the contents of that vesicle can leave. You see this? Examples of when you'll see this in the cell is for like when the Golgi apparatus is trying to ship proteins out. Exocytosis. So endocytosis, if exo is exit, endo is into, in the cell. Endocytosis is the movement of materials into the cell by infolding. So instead of opening a vesicle, we're going to create a vesicle out of the cell membrane. There's different types. Pinocytosis is receptor-mediated endocytosis, and we're looking at liquids with pinocytosis. Just kind of like a wine is a liquid, Pinot, the wine is a liquid. Pinocytosis is the movement of liquids. And it's receptor mediated, meaning that we're looking for specific things. We have receptors on the cell membrane that are looking for specific structures. Okay? And when those materials bind, it allows the cell membrane to create a vesicle, create a pit. And that pit will eventually become a vesicle that goes inside the cell. This is an important way that our cells use to get nutrients and other substances in, and it's also how the flu virus affects us. It's how it's infecting all of our different cells within our bodies. It's using endocytosis. And this is a diagram that kind of helps us to picture that. This is not receptor mediated in this diagram, but it's still showing how the vesicles are forming. So you see you kind of have a depression that's forming here. And as that depression deepens, you get more and more materials in. And eventually that depression deepens enough that you can hold a lot of materials. And this side right here will pinch towards that side right there. And eventually you get an entire vesicle that can be transported within the cell. So maybe we're moving materials that we need in order to build structures or in order to run different cell processes. And we now have that. So pinocytosis. And this is a diagram of it. It's really pretty neat where you have your extracellular fluid here. Fluid. And then we have our cell membrane. And you can see where it's folding in and bringing, like this vesicle's already made, and there's other vesicles that are going to be transported through. So we're bringing materials into the cell. This is receptor-mediated endocytosis. So this is like our receptors are looking for specific materials, and we gather all our materials, and once we gather our materials, we're going to create this coated pit, and then eventually our cell membrane is going to pinch off and create a vesicle. So maybe we're looking for a specific molecule that is needed, all right? And that is how we are creating our vesicles for that. Okay, and this is a diagram of the same thing, where you have your receptors that are collecting materials, your cell membrane is forming that pit, we're going to pinch it off right there, and then you have your vesicle with your material in it. Phagocytosis is cell-eating endocytosis. So in this case, we're looking at solids. Previously, where was it? Pinocytosis is liquids. Remember, the way to remember that is Pinot is a type of wine, and wine is a liquid. Phagocytosis is solid, and this is known as cell-eating. All right, so we're bringing in solid materials. This is how many single-cell organisms will take in food, and also how our human immune system will engulf entire bacteria. So it just kind of gobbles it all up. The vesicles formed in this can be much larger than those formed in pinocytosis, and it can do it in several different ways. It can actually send out cell extensions. So we can actually send out extensions to try to trap a particular substance. And those extensions will eventually, well, let's just draw it as a process. All right, so there's something here. All right, and then we're extending our cell membrane, and then eventually what will happen is we will form a pocket, right? And then now all of a sudden we have a vesicle with that material in. Okay. The vesicle forms will then fuse with a lysosome. So let's get ourselves a lysosome over here. Fuse to a lysosome, and that lysosome, if you remember, has enzymes in it. And those enzymes are going to go into the cell, and those enzymes will break down whatever it is that we just digested. And so that the cell can then take those parts and use it as they need it. So this is basically a diagram of what I just drew. We have our bacteria, our pseudopodia, our cell extensions are being formed so that we can trap whatever it is that we're trying to trap. And in this case, a bacteria. We're going to take it and then send it, infuse it with a lysosome. Now, this is one of the ways our immune cells will protect us from these bacteria, is that they'll actually engulf them. And then some types of immune cells will then take the parts of that bacteria and then display them on the surface of the cell so that other antibodies can recognize that and then attack. It's kind of a vicious process, honestly. This is an example of the cytoplasmic extensions right here. And then you can see they're trying to swallow or cover this structure and then engulf it. I don't know, every time I see this diagram, it makes me think like there's this little bacteria or whatever it is screaming. We got a nose and we got a big open mouth and it's saying, no, don't eat me. That's not really what's happening, but for some reason, every time I see that diagram, that's what I think. All right, so that is the end of cell transport. We covered a lot of material pretty quickly. We didn't get super in depth in it, but what we need to just mainly focus on is the different types of cell transport and what direction. So we need to focus on the types. What direction is movement occurring? And what materials are moved? So if you can do that for all those different types of cell movement, so we've got, what did we talk about? We talked about diffusion, facilitated diffusion, osmosis, active transport, specifically sodium, potassium pump, exocytosis, endocytosis. Let's see what we talked about. With endocytosis, we talked about pinocytosis. We talked about receptor mediated. And we also talked about phagocytosis. Phago. Right? So if you can tell me what direction movement is occurring. If you can tell me what types of materials are moving in, so for instance, facilitated diffusion is going to be charged ions or ions that are too large, or materials that are too large to move directly through. If you can say that about everything that we talked about today, you are in a good place. So if you have any questions, like always, make sure you email me or message me on Remind, and hopefully I will be able to answer you ASAP.